Okay, so today we're going to talk about the cognitive neuroscience of addiction. The Diagnostic and Statistical Manual of Mental Disorders, or the DSM, officially calls this condition substance dependence for just diagnostic and treatment purposes. Substance dependence is considered a chronic relapsing disorder. Scientists have suggested that it involves this addiction cycle shown on the screen here. That's composed of three stages: a binge intoxication phase of drug use and drug taking that then leads to this withdrawal, negative affect, or hangover or crash state. That will eventually give way to this preoccupation, anticipation, or craving state, thinking about taking the drug, wanting to take it again. And then that's supposedly starts the cycle all over again. And this results in the disorder being characterized by a compulsion to seek and take the drug, and then a loss of control limiting intake of the drug, and then an unpleasant syndrome when the drug is prevented. Understanding what causes substance abuse and its consequences is really important because it has wide sweeping effects throughout our society. In the US, substance abuse exerts a toll of more than $700 billion a year in healthcare and crime related expenses. And more importantly, it contributes to the death of more than 90,000 people. Additionally, recent evidence has actually shown that the pandemic may have actually further exacerbated this major public health problem, with about 13% of adults in the US reporting starting or increasing their substance use this last June. All right, so I assume at this point in the course, you all have somewhat of a working understanding of what cognitive neuroscience research is, given the name of the course. In general, it's the study of how biological processes of the brain underlie mental processes and behavior. So, thinking about this definition might give you some ideas about what are some important questions that neuroscientists might be trying to understand regarding substance use. So, I'll just list some broad topics to get us thinking, but this list is not exhaustive, and I'm sure you guys could think of other questions that might be important for science to try to answer. All right. So, one important question is, what are some individual differences between people that might be risk factors for drug use or abuse? Meaning, are there certain phenotypes or types of brain structure or function that make someone particularly prone to using and or abusing substances? Another important question is, how do drugs impact the brain in the short term? How is brain function altered when people are actually under the influence of certain substances or when they're in withdrawal? It's also important to understand the long term impacts as well. How does the brain change after repeated use over and over again? Another important question neuroscientists might be trying to address is, how do these changes contribute to the escalation of the addiction pathology? How do they impact everyday functioning or lead to mental health problems or other negative outcomes like dropping out of school or accumulating financial debts? These are all important questions. Throughout this presentation, we're just going to talk about some cognitive neuroscience research findings that have begun to illuminate answers to some of these important questions. Okay, so first, before we get into that, let's just talk about some fundamental reinforcement processing mechanisms that can be dysregulated by repeated addictive drug use. Positive reinforcement is a psychological term that you might have heard before. It refers to any consequence that's following a behavior that increases the likelihood of that behavior occurring again. So, this concept originally stems from research in animal models, but since then, there's been a lot of behavioral and brain research examining positive reinforcements in humans as well. So, the neurocircuitry supporting reinforcement processing and reinforcement learning is extremely important for adaptive human behavior and habit forming. Um, it's what allows us to learn what behaviors we need to do to get the things we want and need. For example, a toddler learning to drink juice out of a sippy cup for the first time. So, juice and water can be reinforcing, especially when you're really thirsty. They can release the neurotransmitter dopamine, which in certain contexts can be thought of as the brain's learning signal. This same powerful learning and habit forming brain circuitry can be thrown off balance in certain vulnerable individuals by addictive drugs. or other particularly potent positive reinforcers like yummy food, sex, or gambling, or by intense negative reinforcers like extreme or chronic stress. These potent drug reinforcers can unbalance dopamine learning signals because they cause these unnaturally high and quick spikes in dopamine. Research has shown that learning from experience to perform appropriate behavioral responses is supported by dopamine signals coming from nuclei in this midbrain area that arrive in the striatum. Specifically, neurons in the ventral tegmental area and the substantia nigra, which are relatively small nuclei in this midbrain area here, send projections primarily to the nucleus accumbens region in the striatum or the ventral striatum, and they release dopamine there. They also send dopaminergic projections to the dorsal striatum, amygdala, hippocampus, and prefrontal cortex. So, simplistically, dopamine projections to the striatum have been linked with reward and motivation, and dopamine projections to the prefrontal cortex have been linked with self control and working memory. All right, so here's an even more in depth look at dopamine driven learning process. This was in your reading for this week, and it could help us understand how the toddler learns to drink out of her sippy cup. So, after neurons in the ventral tegmental area and substantia nigra, those two small midbrain nuclei we were just talking about, release dopamine in the striatum, the dopamine has to bind somewhere in the striatum. So, it primarily binds to medium spiny neurons. There are two classes of these medium spiny neurons in the striatum, and they differ significantly in terms of their dopamine receptors. Medium spiny neurons in what is called the direct pathway, shown here in red, express D1 type dopamine receptors, which enhance dendritic excitability and glutamatergic signaling and promote behavioral action. Whereas medium spiny neurons in this indirect pathway here, shown in blue, express D2 type dopamine receptors, which are thought to suppress behavioral action. So, when an action is performed that results in a better than predicted situation, like the toddler tips her sippy cup in a certain way and she finally gets some juice in her mouth instead of on the floor, dopamine neurons fire a burst of spikes, which is likely to activate the D1 type receptors on the direct pathway neurons and facilitate immediate behavioral action and potentially lead to neuroplastic changes that could make our toddler more likely to select that particular cup tipping action in the future. In contrast, when the result of an action is worse than expected, like the toddler tips her sippy cup a certain direction and the juice just spills all over the floor, and then the toddler's parent comes and takes the juice away, dopamine neurons are inhibited, reducing dopamine, which is likely to inhibit D2 type dopamine receptors in the indirect pathway, and then suppress immediate action and potentially the suppression of unsuccessful cup tipping actions in the future. So, overall, this mechanism is thought to support learning to perform appropriate behaviors based on your experience. Here's another example of some of the nuance of how this is thought to guide learning. Specifically, there is evidence of two populations of dopamine neurons in the ventral tegmental area that differ in the nature of their predictive, like firing code that is used to learn. So the first type are these motivational value coding dopamine neurons. They're thought to encode prediction error signals, and they have strong inhibition by the omission of rewards and mild excitation by the omission of aversive events. So let's get into this. We see here these dopamine neurons spike in response to a predictive reward cue, a cue indicating they're about to get a reward. In this case, it's juice. Um, and then we see that they have a dip, they're inhibited in response to a cue that indicates they're about to get a puff of air in their eyeball, which is very aversive, not a good feeling. It's a punishment. And then here we see the response to the reward and the air puff when they weren't expecting it, when they didn't get a cue to indicate that it was going to happen. Again, we see firing for the reward, inhibition for the punishment. And then this is when there's an unpredicted no outcome. So when you were expecting to get a puff of air in your eye and you were like, oh no, here it comes, and you didn't get it, we see just this little bump like, yay, it didn't happen. And then here we see when you're expecting to get some juice, when you were hyped for it, and then you didn't get it. We have a little bit of an inhibition of dopamine here because we're like, poop, I wanted that juice. It's the disappointment inhibition. And then here we see the salience coding dopamine neurons. So this is interesting. They're clearly doing something different to the same stimuli. They're, regardless of the valence or whether it was a good or bad thing, they are being fired by these cues. And that they're thought to just encode things that are salient, that are important, regardless of whether they're good or bad. So, this activity is thought to guide associative learning about things that are happening to you. So, overall, the point is these dopamine signals are extremely important for your everyday behavior. However, like I mentioned at the beginning, this mechanism can be dysregulated by drugs like nicotine. Whether directly or indirectly, all addictive drugs have the power to cause these large increases in dopamine from the ventral tegmental area in neurons that project primarily to the ventral striatum and beyond that we talked about earlier. So, nicotine releases very large amounts of dopamine in the ventral tegmental area. And after being repeatedly exposed to nicotine over and over again, the brain eventually adapts by reducing dopamine responses. We'll talk about why this happens on the next slide. As the brain changes, a person then needs nicotine to keep brain function stable and avoid withdrawal. So let's unpack why this happens. This is an extended version of the opponent process model that's been used to explain this process. The idea is that in general, the human body and human brain are always trying to maintain homeostasis, represented by this flat line here. So when a drug increases dopamine outside normal levels, represented by this bump here labeled as the A process, the brain will enact appropriate counteractive physiological B processes to balance out the A process. In this case, that might mean reducing dopaminergic responses to positive reinforcement because nicotine is always hitting the brain with really high dopamine spikes. These B processes are also thought to explain the buildup of drug tolerance over time. So, after repeated use of a drug over and over, the transition to addiction is thought to stem from this transition to an allostatic state. That means that because our reaction to a drug after repeated use decreases each time because the brain and body are becoming desensitized to its effects, the B processes, shown here, never returns to their normal original homeostatic level before drug taking begins. We also see these B processy dips are getting lower and lower with each drug experience. And the return to homeostasis, circled here in yellow, is getting lower and lower as well, thus creating a greater and greater allostatic state. Here, when the baseline homeostasis level is shifting more and more. So this is how drugs are thought that they might change the brain over time. So examining altered brain function, particularly altered reward processing brain function, is really important question in cognitive neuroscience. When considering altered brain function related to drug use, we can categorize alterations into two broad categories, namely state-like aspects and trait-like aspects. State-like aspects refer to how the brain is altered during drug withdrawal or drug use. So these brain processes change depending on whether an individual who is substance dependent is in drug withdrawal or not. The other category is trait-like aspects. These brain alterations should be the same regardless of whether someone is in withdrawal or not, regardless of what state they're in. But they differ between substance-dependent individuals and control, non-dependent individuals. When considering these trait-like aspects, another important issue to always keep in the back of your mind when doing research or reading research is whether brain function among substance-dependent individuals that's altered compared to controls was caused by their repeated drug use, or whether they already had differential brain function before they started using that made them more likely to start using and abusing the drug in the first place. So it's a bit of a which came first, the chicken or the egg problem, and we'll discuss that more in a bit. So first, let's talk about state-like aspects, specifically withdrawal symptoms. I'm going to use nicotine as an example addictive drug in this and following slides, but note that a lot of what I explain about nicotine applies to other addictive drugs as well. So, nicotine withdrawal often has somatic, cognitive, affective, and reward processing symptoms. Some nicotine withdrawal symptoms can be pretty brutal. They include anxiety or nervousness, lack of concentration or brain fog, like trouble focusing on work or getting things done, irritability, like being short tempered with friends and family, insomnia, like trouble sleeping, and lack of pleasure from everyday experiences, like not enjoying the things you normally would. It's often called anhedonia. These withdrawal symptoms are a major barrier to quitting smoking. As we talked about before, the motivation to relieve these symptoms is strong, and that is thought to perpetuate this cycle going back for another cigarette. Even when someone wants to quit and they're trying a quit attempt, they will often experience severe withdrawal symptoms for a week and often longer. So, understanding the changes in the brain that may drive these symptoms could be really important for improving and developing smoking cessation or quit aids, interventions, or medications. So, how exactly is nicotine changing the brain? Functional magnetic resonance imaging studies have been used to address this very question. I'm going to now talk about some findings from a series of fMRI neuroimaging studies, and I'm not sure if you guys have already gotten some background on magnetic resonance imaging, but the idea is that a participant will lie in an MRI scanner for usually about 30 minutes to 2 hours and have their head scanned so we can construct an image of their brain. I won't get into the physics of how this works, but the basic idea is that the MRI is non-invasive, so it doesn't pose any health risks to participants, and it uses strong magnetic forces, way stronger than the Earth's gravity, and radio frequencies to gather information about our brain tissue so that a three-dimensional image of our brain can be constructed. The MRI is also able to capture information about where in the brain tissue is getting oxygenated blood flow, which we use as a proxy measure for brain activity or brain function. So I'm going to talk about some findings from an fMRI dataset of participants that were split into two groups, dependent cigarette smokers and non-smokers. These participants were scanned multiple times on different days while they were in different drug states. Specifically, the study involved the administration of two different smoking cessation aids or medications to help quit smoking, with corresponding placebo conditions. This allowed us to examine brain function when smokers were in nicotine withdrawal and when they were given a transdermal nicotine replacement patch, which relieved their withdrawal, and when they were administered a regimen of vernicline pills, which is another smoking cessation aid. It only partially relieves nicotine withdrawal, though. It's a really interesting drug. Its commercial name is Chantix. You might have seen one of their commercials on TV. What's interesting about this study design is that transdermal nicotine replacement patch is a full agonist of nicotine, meaning that it provides the same action that nicotine from a cigarette might when it binds to nicotine receptors. You see here, this little yellow thing is supposed to be nicotine. This is a nicotine receptor firing dopamine neurons. That's dopamine being released. Um, this is when smokers are in withdrawal. They don't have nicotine. No nicotine is binding, not that much dopamine withdrawal. And then this picture, these little pink things are representing vernicline. Vernicline is a partial agonist, partial antagonist, meaning it only produces about 50% of the action of nicotine at nicotine receptors, but it also blocks 50% of nicotine's action. That's why it's used as a smoking cessation medication, because when you're taking it, it makes the nicotine from a cigarette not really bind in your brain. So it takes up little spots on these receptors, doesn't let nicotine bind, but it only has 50% of the action of nicotine. Um, so what that looks like here is nicotine, shown by this red line, is fully different than the placebo condition, shown by this gray line, in its effects. But when vernicline is added on top, whether you have nicotine or you don't have nicotine, you're in withdrawal, it should vernicline should bring you to this middle point of nicotine action, this 50% action here. So I tell you all this so you can look for this expected pattern of drug effects in the coming slides, because this is what we are expecting to happen. Okay, so we can check that these drugs are doing what we would expect them to do in smokers and non-smokers by looking at a physiological measure, like heart rate, and a psychological or cognitive measure like attention. So it's pretty well established that nicotine increases your heart rate, whether you're a regular smoker or not. And sure enough, we see here a similar pattern of drug effects that were predicted both in our dependent smokers and in our non-smokers on this heart rate dependent measure. It's also well established that when dependent smokers are in nicotine withdrawal, they have difficulty paying attention and do worse on tasks that require attention. So here we examine the reaction time in a task as a measure of their task attention. So here in smokers, we see the expected pattern of drug effects on their reaction time attention measure, but we don't see the same pattern in the non-smokers. Anyone know why this is? The idea is it's because non-smokers are not in withdrawal. Presumably, when they don't have nicotine, they're not experiencing nicotine withdrawal because they're not addicted. So changes in attention versus changes in heart rate seem to be specific to the effects of nicotine withdrawal and change when drugs relieve nicotine withdrawal, but they're not just an effect of the drug absent of their nicotine withdrawal relieving effects. So now that we understand the effects of these two drugs, we're in a better position to understand their effects on brain function. All right. So the first results I'm going to talk about are on measures of functional connectivity in the brain. One of your readings for this week focused on this measure of functional connectivity as well. So let's just do a quick review of what functional connectivity in the brain is. So the study we're going to talk about did a seed-based connectivity analysis, meaning that they extracted brain activity, represented by this top jaggedy line here, from a particular seed region, for example, this green circled brain region here. And then they want to examine how similar this extracted brain activity looks to the brain activity extracted from all the other parts of the brain, represented by this bottom jaggedy line here. In particular, this study was looking for places where the jaggedy lines were doing the same thing at the same time, meaning they went up at the same time and they both went down at the same time. If this correlation between the two is statistically significant, we would say our seed region is functionally connected to that part of the brain. Across the 8 minutes of the scan, the two regions were doing the same thing at the same time. Okay. This study shows certain brain function that might underlie some nicotine withdrawal symptoms. The first author is Dr. Matthew Sutherland. Dr. Sutherland is actually a professor here at FIU, and he's my advisor. So as we talked about before, it's really important to understand biomarkers of withdrawal symptoms because they're really unpleasant and make quitting smoking hard. So this study was specifically looking at functional connectivity of two brain regions, the amygdala and the insula. The amygdala is shown by this little red dot here, and then the insula is this big, colorful, spinny thing here. So this study found that the strength of functional connectivity between the amygdala, this little green seed region here, and the insula was downregulated or decreased by both nicotine and vernicline in smokers. This study also found that functional connectivity between the insula, green region here, and the posterior cingulate cortex was decreased also by these two drugs. So here, the amygdala was our seed region, and it's showing its functional connectivity to the insula. And here, the insula is our seed region, and we're seeing the functional connectivity to the posterior cingulate cortex. But these effects were only observed in smokers, not non-smokers. So we see here a significant difference between placebo and nicotine in the smoking group, but not significant difference between the two conditions in the non-smoking group. Again, here for the posterior cingulate cortex, significant difference between placebo and nicotine in the smoking group, non-significant difference in the non-smoking group. So hopefully our setup background info makes it clear that this suggests that these effects were associated specifically with nicotine withdrawal state, rather than the effects of the drugs absent of the nicotine withdrawal relieving effects. So that heightened functional connectivity between the amygdala and the insula, and between the insula and the posterior cingulate cortex, was associated with being in nicotine withdrawal. This heightened functional connectivity between the insula and the posterior cingulate cortex during withdrawal is really interesting because the posterior cingulate cortex, this region here, is a major hub of a functional brain network called the default mode network, shown here in blue. So the default mode network is part of this tripartite, meaning three, framework of psychopathology. The framework suggests that dysregulation of the balance of these three large-scale functional brain networks that are often seen can explain neurocognitive symptoms of a bunch of different disorders or conditions. So these commonly observed functional networks usually include the salience network, shown here in green, the default mode network, and then also this task positive network or activation patterns commonly seen during cognitive tasks that involve monitoring or interacting with external stimuli, like a video game. Um, and the default mode network is a brain network that's commonly seen when people are just chilling, letting their mind wander, when it's suppressed when people are doing tasks that require attention to be focused on external stimuli, but it's activated during just like mind wandering, daydreaming, internal thoughts about your internal state, and memories. So, as you can imagine, turning down a brain network that's involved in daydreaming when you're trying to do a fast paced, cognitively intensive task like a video game or your homework or something might be helpful and make you better at the task. The insula is a major hub of the salience network, shown here in green. This network is thought to monitor both internal states, like what the default mode network is involved in, and external stimuli, and then turn up or down the default mode network or other task positive networks, depending on what might be the most advantageous to pay attention to at the given moment. So all that being said, if we see this increased connectivity between the insula in the salience network and the posterior cingulate cortex in the default mode network during nicotine withdrawal, this may reflect smokers' difficulty concentrating during nicotine withdrawal, possibly because they're having trouble turning down their thoughts associated with the default mode network, like about their internal state, maybe about how they don't feel super great, or maybe they have a headache because they're in withdrawal, or how they could really go for a cigarette right about now. Alterations in the balances between these networks is hypothesized to both characterize various psychopathologies, but also a drug withdrawal state. Specifically, during nicotine withdrawal, shown up here, smokers are hypothesized to show this heightened resource allocation from the salience network to the default mode network when nicotine is administered down here. More resources are thought to be allocated towards from the salience network towards these more task-related brain networks, possibly facilitating paying attention. So we see this actually play out with real data from this study from 2014. This study was cool because it showed this dysregulation in these brain networks when smokers were in a withdrawal state, but it also related these measures to performance on a working memory task. Okay, so we've been talking about nicotine withdrawal state-like effects. But even withdrawal is a multifaceted state. There's a lot going on. So another study found that this emotion-related amygdala activity here is heightened during nicotine withdrawal. this might be related to the anxiety symptoms that are often seen during nicotine withdrawal, given that this amygdala activity was in response to emotional phase stimuli, and because there's extensive prior research linking the amygdala with fear processing and anxiety. Another brain region called the habenula, which is known to put the brakes on dopamine in the striatum, was increased in response to rewards during nicotine withdrawal. And this alteration was related to the degree of cigarette craving. So these studies suggest that altered amygdala and habenula function also likely contribute to characteristics of this withdrawal state. So these brain alterations associated with state-like withdrawal aspects may begin to show us some of the neural mechanisms that drive nicotine withdrawal symptoms like difficulty concentrating, anxiety, and craving, or anhedonia, meaning not liking things you normally would because this brain activity was in response to rewards. So, as I mentioned before, trait-like aspects in addition to these state-like withdrawal aspects are important to examine. Specifically, one of the studies from my lab here at FIU displayed striatum responsivity to rewards that was blunted in smokers and it was related to their nicotine addiction severity. But importantly, this altered striatum response seems to be a trait-like effect because it didn't change across the drug conditions. Whether smokers were in withdrawal or whether they had received nicotine, they still showed the same blunted striatum response compared to the non-smokers. Another trait-like effect that's been shown pretty consistently, here it's shown among cocaine and meth users, but it's also been thought to characterize various addictions, is reduced levels of dopamine receptors in the striatum, shown here, and reduced dopamine release in the striatum. As we talked about earlier, dopamine release in the striatum is important for reward processing, and reward learning, which we went into earlier, is really important for just everyday functioning and learning. Here, you see a 3D rendering of the striatum and its different subregions inside a glass brain to kind of give you an idea of where this important brain region is in the brain. So these reward processing alterations have been displayed in fMRI neuroimaging studies of dependent cigarette smokers. Largely, these studies have consistently shown blunted striatum responsivity to non-drug rewards like money or positive feedback like whether you did well on a task indicated by this green smiley face. Interestingly, this blunted striatum response has also been shown among adolescent smokers that have a pretty limited lifetime exposure to nicotine. They're only teenagers. If they are smoking, they haven't been smoking very long because they're still relatively young. So you see here, this is the non-smoking comparison teen subjects, and then that's their striatal response to non-drug rewards. And then we see a much less activity among the smokers. So this might bring up the question of whether these trait-like blunted striatal reward responses that we keep seeing across a bunch of different data sets among smokers are always like lower, are in response to repeated drug use, or if they've always been lower even before drug use started, and might be making people more likely to start smoking or using drugs to compensate for this low reward responsivity. And that we don't know that for sure, but just given that we see it so pronounced among a group of smokers that have only been smoking for like a year or two because they're teenagers. So this brings up the chicken or the egg problem for addiction neuroscientists. Are the changes we observe pre-existing before use and leading to use, or are they caused by the drug? Luckily, the National Institute of Health has funded a very large scale multi-site study called the Adolescent Brain and Cognitive Development Study, or the ABCD study. Got a great acronym. It was designed to be able to address this very problem. The project started in 2018, and it's being conducted at 21 different research sites all over the US, including one here at FIU. This project starts collecting a bunch of data about the development, health, and brain of about 10,000 9 and 10-year-olds. And then it follows these children through their adolescence and development into young adults. So this study will have data on these individuals before and after they start using drugs. Thus, it can examine risk factors for using drugs as well as what effects are specifically caused by drugs that didn't exist prior. This is possible because it has a longitudinal research design, meaning we are collecting data from the same individuals over multiple time points at different time points, so we can observe changes over time and over development, and presumably over the shift from first drug trying experimentation to escalating regular use. All right. So I'm getting to the end of all the info I wanted to chat with you guys about today. But before we start a discussion, I just wanted to show you some interesting stats regarding the rapid rise in e-cigarette use among teens that you might have heard about in the news. So, while cigarette use among teens has been declining for about the past 15 years, which might have been brought about by various reasons, including aggressive public health policies and regulations regarding smoking, like smoking in restaurants and in airplanes is pretty much completely prohibited in the US now. And that wasn't even the case when I was a kid. However, electronic cigarette use or vaping has skyrocketed, particularly among young people in the past couple of years. It's been deemed an epidemic by the Surgeon General. These new generation vapes are being marketed to teens, and they can deliver nicotine concentrations as high or higher than a cigarette can. So it will be interesting for research to examine whether many of the findings that we observed among dependent cigarette smokers that I've been talking about today will show similar effects among heavy vapors, or if similar or new risk factors predispose someone to vaping nicotine or cannabis. So this is all something stuff that my advisor is currently working on here at FIU. So it'll be interesting research to tune into in the next couple years. All right. Thank you all so much for listening. Let me know if you have any questions. We can chat about more.